So I'd like to thank the organizers for the opportunity to be here today, the invitation. And I want to say it's quite an honor to be included with this session. And I'm kind of excited to be here talking with this group of people who are taking things from basic science and moving it into real life application. Because I come at this from the basic science translational position. You can probably tell because I'm not wearing a coat that I'm not an MD. I'm going to talk today about potential interference with recovery after spinal cord injury, things that might happen spontaneously, or some of these therapies that we're using, there might be some interference with this. And interestingly, for those living with spinal cord injury, a lot of it is stuff they don't even know about because there's lack of sensation. And so the basis of all of this is going to be something I call sensory pathologies. I do have to offer some disclosures. I'm part of two companies that are commercializing technologies from the lab. My role there and those technologies have nothing to do with today's talk. So fundamental message I want to get across up front so that we can understand the rest is that there is circuitry below the level of the spinal cord injury which was completely or mostly intact, and it still functions. That's going to become really important. What's an example of that? Well, we've all probably been here where we're getting the patellar tendon reflex, stretch reflex at the doctor's. You don't need a brain for this. All spinal cord. Well, what about something more complex? Walking, running. A lot of the circuitry that controls these functions is resident in the lower spinal cord. If there's a spinal cord injury that completely cuts off communication, a lot of this stuff can still happen. And so it's still functioning. In fact, as has been mentioned already, that circuitry, especially the sensory input to the spinal cord, is required for these successful therapies that we have. Treadmill training, epidural stimulation. So that circuitry is required for these things to be effective. And it is necessary that the sensory input is part of that, even if the sensory input is not perceived as a sensation. Now, on the downside of this, as has been mentioned a few times today, there's autonomic dysreflexia. This circuitry exists before anybody's injury. It's simply a loss of control from the brain. And unfortunately, it's all too common in our folks who are living with spinal cord injury. It's almost a spinal cord injury specific condition. But really importantly for today, it is a nociceptor dependent process. This is driven by the pain receptors that are still functioning after a spinal cord injury. So a lot of us have probably experienced this sort of thing. You're walking along, maybe in your house, walking on a beach. Anybody who's got kids has probably walked along in the middle of the night and stepped on a Lego that's in the middle of the hallway. I know I have. Well, as is exemplified here, things change. So as we saw Mr. Bean running earlier, I said a lot of this circuitry, it's automatic. The motor programs are automatic. They're in the spinal cord. These folks didn't keep walking. What'd they do? They stopped putting that foot down on the Lego and they put the other foot down to keep their balance. That's in the spinal cord. So a lot of that circuitry, it's there and it still functions. The real thing I want to get across here is that the pain receptors that are activated by that Lego, they override the motor programs. They have override authority for the spinal motor programs. Important point. Now, those pain receptors in a non-spinal cord injured condition, it takes a lot to get them to go. They're high threshold. It takes a lot of strong pinch or heat. It takes a lot to get them to go. After spinal cord injury, they have a surprisingly low threshold for activation. This is a big change. In fact, some of them have such a low threshold that they're activated spontaneously. And this can create a lot of problems. So first part of this is motor circuitry exists in the spinal cord. That motor circuitry and the sensory input is necessary for the currently successful therapies that we have. The nociceptive reflex circuitry, the pain reflex circuitry, is also intact after a spinal cord injury. Those nociceptive reflexes can interfere with the motor patterns that someone might have or might be trying to train and improve. And the nociceptors themselves that drive those reflexes, they're sensitized. They're more easily activated. So, great. What does all this mean? I'm going to show you some data here. This is work that's been done in rats with myself and my colleagues. It's a locomotor scale. At the top, 21, before injury, these rats can walk along fine. We give them an index number of 21. After their moderately severe spinal cord injury, that goes down to 0. They can't really do anything. Over the course of the next 4 to 6 weeks, they recover to a very predictable and very regular level of locomotor capability. Now, I happen to work across the hall from a colleague who's really brilliant. I've been able to learn a lot from him. He has started doing things in rats that we just do in people for clinical necessity. He has put rats in wheelchairs. In this particular study, we did therapeutic stretch on the rats. Much like a lot of the folks who are in chairs and have been dealing with this all their lives have to get on a regular basis. We even had the PTs come over and the bioengineers to make sure that we were applying to the rats a very, very similar sort of stretch that people get. When we give that stretch, the locomotor capacity that those rats had regained is gone. Completely dropped off. It happens fast, it doesn't take much stretching for it to happen, and it's long-lasting. You stop the stretch, and over the course of the next couple of weeks, the animal's abilities come back, not always the same. This was really disturbing a little bit to us, and well, we're like, if this is actually happening in people, and it looks like probably it is, some of the PTs have gotten involved with this, and it looks like this might actually be happening in humans as well. So we asked the question, why? What is it? What about that stretch is causing this suppression of locomotor function? And I said to my colleague David, I said, I think it's the nociceptors. I think that it is this override of the motor program that's happening, so let's do the same experiment in animals in which we have depleted the nociceptor population. And if you get rid of the nociceptors, you can give them that exact same stretch, doesn't hurt their walking at all. And so what that tells us is that the nociceptor activity is required for the stretch-induced suppression of locomotion. And it looks like, as I mentioned, it looks like this is probably happening in humans over the course of the next year. I think we'll get some papers coming out on this. So then I asked, well, what is it about the nociceptors? Why are they being sensitized after spinal cord injury? A lot of them are not actually injured themselves. These sensory neurons, generally they're okay. Why are they so much easier to activate? Well, there's the spinal cord injury, it's an inflammatory environment. The cerebrospinal fluid that surrounds the spinal cord, it has a lot of inflammatory mediators inside of it. Well, the spinal sensory neurons are encased in that same dura. They're bathed in that same CSF. Maybe that's what's doing it. So we did an experiment where we took sensory neurons from a completely intact, naive rat, put them in a dish so that we could ask a question of, how easily are you activated? And there's a normal range of that. And then we took some cerebrospinal fluid from a pig that had been injured 1 month earlier, a gift of Brian Quanup and I-Cord, and we just sprayed a little bit on our sensory neurons. And they were significantly easier to activate after just a little bit of CSF acutely sprayed onto them. And this effect was only for the nociceptors, not the sensory receptors for your hairs or stretch or anything like that, just the pain receptors. So again, what are the implications of this? One, this, to speak to a lot of the things that are happening here, maybe trial design. It's possible that if this were validated, beefed up a little bit, we could get fluids from our participants, and it's possible that this could help parse our populations so that we can get some more information about them. Therapy development. One of the principles here is that there's something in the cerebrospinal fluid that actually sensitizes those sensory neurons. Maybe that's a therapeutic target. Something else that we do need to be concerned with in terms of nociceptors and trial design, et cetera, is that nociceptors are different, whether they come from men or women, and this is true from mice up to humans. It's a brand new paper from an excellent researcher, Frank Petruska, and this is something we're going to have to bear in mind. All that's been a bunch of bad news. There's also a little bit of good news. I want to finish with that. Levels of activity can modulate some of the spinal cord injury-induced changes in sensory neurons. So I told you nociceptors are sensitized by stuff that's in the cerebrospinal fluid after an injury, and that's an extrinsic factor that makes them easier to activate. Well, they also have changes in the genes that they express. So you do a spinal cord injury, the sensory neurons that are nociceptors, they have a big change in what they're expressing. Some of it goes up, some of it goes down. Well, if you give added activity, you put them on treadmill or give them shallow water walking, things like that to induce added activity, those spinal cord injury-induced changes, they get reversed for many, many of those genes. And it's not just any random genes. In fact, a lot of the pain-related genes, the ones we know are a part of pain, they're actually reversed too by activity. And I found this pretty interesting because obviously a lot of things change after spinal cord injury: metabolism, muscle activity, et cetera. So we tested, what are the gene changes in sensory neurons, liver, muscle. Activity induces the greatest number of reversals, like basically a therapeutic direction, in sensory neurons, more than liver or muscle. So that's a good sign. End of story here. Sensory input below spinal cord injury, even if it's subliminal, it's still coming in. And the effects that sensory input can have are real, even if folks can't perceive the pain. And so effective interventions and trial designs should probably not be agnostic to this, not saying that they are, but I think it's something that can be incorporated and should be. Again, thank you for the opportunity. Thank you for your attention. The names of some of my collaborators and folks who did the work, funding agencies, and places I've trained. So thank you very much.